Thanks a lot for the nice introduction, Angela. I'm very pleased to give this talk before you today, and hopefully that will be some pharmacology, clinical pharmacology. I'll do my best, but as you can imagine, both are people from all sort of different disciplines need to be involved in moving this particular area of the field forward. And this is a summary of what I would like to cover, where we currently are and why that is unsustainable. And then I'll move on to talk about new approach methodologies and how some of them are potentially applicable for pregnancy studies. And then I'll conclude with this important point. So this is where we are at the moment. We do all of these studies, preclinical studies. We move on to phase 1, phase 2, phase 3. It is only after there is evidence of some level of success in phase 2 studies that pregnancy studies start. But these are in rodents and non-rodents, mainly rat and rabbit are used for this type of studies. So why is this not sustainable? We're very familiar with this drug attrition problem that a company starts out with 10,000 molecules in discovery, and then they end up with maybe one at the approval stage. But when you look at pregnancy, the situation is far worse. Because if you start out with, for instance, 110,000 molecules in discovery, and you come out with 10, it's only for one out of those 10 that we have sufficient information regarding whether they are safe to use during pregnancy or not. And the reason is very simple, because the animal models that are currently used to assess drug safety, they do not adequately represent human pregnancy. So we need timely human pregnancy relevant information before we can begin to have timely recommendations. Now, just to emphasize the reason why the current situation is not sustainable, I'll use these popular drugs to illustrate. This is a summary of developmental and reproductive toxicology studies, 3 different segments, and those are the different types of studies that are conducted during these studies. Now, when you look at lenacapavir, cabotegravir, Glen and pibrenthevir, what is the summary of the available information in prescription information for them? That's the summary, but then, so these are the data from prescription information for all of them, but what do they tell us? For pregnancy, it tells us that we're not adequate, we're not certain whether this can be safely used during pregnancy or not, and it's the same for the other 3 drugs. We hope to get more information from pregnancy exposure registries, which exist for LEN or for cabotegravir. There is a planned study starting in November 2025 for JP. So that's the summary of what we have, even with these really popular drugs. Now, let's look more closely at integrase strand transfer inhibitors, CAB, DTG, ratigraphyl, and bictegravir. This study was published recently, just a couple of months ago, and it was conducted in a rat model. It's a developmental and reproductive toxicity study. And here is the summary of what it says. And that might be surprising for you, because even for cabotegravir, it shows evidence of poor pregnancy outcome across all the different types of assessments that were conducted. But of course, we know these models are not adequately representative of what we expect to see in humans. We do have pregnancy exposure registries for these particular drugs. And for prescription information for CAB, ratigraphyl, bictegravir, no evidence of harm, but we need more data. For DTG, you'll be surprised again, the prescription information even to date still says, well, it looks like there is a problem with pregnancy. But of course, we know that what we need more reliable, globally acceptable models that everyone across different stakeholder groups can accept. And then we can use those to inform prescription information that we then use. So the new approach methodologies that I want to talk about, these are innovative methods that do not rely on the traditional animal models that I've mentioned previously. They could be computational and they could be in vitro human relevant models, including organoids and organ-on-chip. And these are the advantages. They are better representative of human physiology. They help us to move away from animal testing, and they are much more efficient for the reasons that I cited on this slide. So before I move on to tell you about the in silico one that I want to focus on, just to call attention to this particular study that showed that for all the different areas that were evaluated across 420 papers, the areas where we have the most information regarding pregnancy, they are not a lot. But antivirals, particularly HIV, tend to be one of those areas that are highly studied, particularly because of impact studies. For HCV, less information is available. In fact, for the number of drugs that you see there, not even one of them assess PK during pregnancy. And I look closely at antiretroviral exposure during pregnancy. This is a summary of all the studies that I could find for what happens during pregnancy. And you can see for almost all the HIV drugs that we currently have, clearance is increased during pregnancy and exposure is reduced. Here is a summary of pregnancy PBPK models that have been published since 1990. And you can see very clearly that there has been a very rapid increase after 2015. This year, we're already catching up with last year, so we'll probably surpass the number of studies that were published. And this is the first new alternative methodology that I want to touch on. The model that I will be summarizing in this talk, the PBPK model, is one developed by Shaquille in my group. Maybe you might remember this particular study published also recently, where Shaquille modeled the initiation of carbapenem during pregnancy. And the summary of the data from that study is the fact that for ripivirine, exposure is below target in a significant proportion of patients. But this was not to steady state, it was at initiation. So he has gone ahead to look at what would happen if these drugs were actually dosed to steady state. And here is the summary, that for in 2015, this year, he presented this at CROI. And this was also discontinuation, but it was after steady state. And it showed really clearly that it took a long time for these drugs to be cleared from the system. For cabotegravir, it continued to be detectable in the system even during the postpartum period, even though the discontinuation was right at the start of pregnancy. So Shaquille has now taken this forward to try to see what if this was continued and not discontinued after pregnancy was detected. So he came up with this steady state cross-gestational PBPK model for long-acting therapeutics, because the key challenge is the fact that you need several models connected together. And you're talking about a long-acting therapeutic. One of the key things that was modeled in this particular area was trying to remember the fact that what helps the long-acting delivery of these drugs is their flip-flop kinetics. It's injected, it stays at the depot site and gradually released, and that controls the long-acting properties. But one thing to remember is at the time of the next injection, you still have some residual drug at the depot site. And even after that, at the time of the next injection, you still have some residual drug. So you got to consider how long it takes for that to reach steady state. So he used some modeling tricks to connect pre-pregnancy model with first trimester, second trimester, and the third trimester model. And he used that to investigate what happens if carbapenem was continued during pregnancy. And he came up with this, that for cabotegravir, there is a decline in exposure during pregnancy, but not as bad as one would have expected. A little bit different for ripivirine. There is a sharp decline in exposure, But the key that I really want to point out is the fact that for both drugs, exposure was predicted to stay above the expected minimum threshold for efficacy. And this is quite important, but of course this is an unpublished work and we still need to understand more, especially as we see more data coming out of ongoing studies. Now move on to human pregnancy relevant in vitro models used in developmental and reproductive toxicity studies. These are the different ones that I want to touch on, not all of them, but I'll touch on a few. And this just goes to show that there are so many tools currently available, and we just need to begin to think in a different way and see how we can answer the question, can some of these potentially be used as a replacement for animal testing? And the first I want to talk about is the placenta-on-chip models that have been developed over the years, and the development has really picked up. And what I have here is what the maternal-fetal interface looks like during the first trimester. During the first 2 weeks, those are the things taking place, including implantation, but particularly I want to call out the trophoblast differentiation. There are the major cell type is cytotrophoblast, and that changes to syncytial trophoblasts and extravillous trophoblast during this period. Subsequently, until week 12, lots of other things take place, including spiral artery remodeling, as you can see in that image. And subsequently after that, further maturation takes place. Since 2016, some of these models have been published, and it's one of the key areas of work in my group. Most of the work that we do in this area is done by Carolina and Elaine, whose pictures you see there. And what we try to do is to co-culture different placenta cells within organ-on-chip and then expose them to the kind of dynamic flow that you have in vivo. And that has really helped us to faithfully recapitulate some of the biology of the human placenta. And it has potential application to model some aspect of the human biology from the time of implantation all the way to the different stages of fetal development. And this is very important because some of the outcomes that we see in pregnancy, as well as the subsequent development of the baby, are actually things that involve placenta perturbation. So most of the time it's not just about how much of the drug gets to the baby. Sometimes not sufficient amount of the drug crosses the placenta, and yet placenta outcomes are really poor. Maybe you might remember the story of the protease inhibitors. When you look at the fetal exposure, very minimal. In fact, for some of the protease inhibitors, you could hardly detect them in the cord blood. But the outcomes were not as good as NNRTIs with extremely high levels of fetal exposure, showing that the placenta is really a major organ, and it's been associated with some of the congenital anomalies that are also common. So that is about the placenta-on-chip, and the ongoing work in this area is also informed by PBPK modeling. So what we do for the in vitro work that we do, we use PBPK modeling to predict how much of the drug the particular cell type we're interested in is exposed to, and we use that for the in vitro work that we do. So the next one that I want to touch on is vascularized organoids and their potential application for fetal organ modeling. This is a very recent work that was published in Science, and the importance of this is really in how it has advanced the field of organoid research. Because over the years, a major disadvantage is the fact that organoids are not perfused, and it can be quite difficult to model all the different aspects of biology that you're trying to model. But now, in this particular paper, what they did was to use a cocktail of vascular-inducing growth factors, and this enabled the development of multi-lineage cardiac and hepatic organoid, as you can see in this image. And they were actually able to show that it shows the cardiac cells and the hepatic cells in this particular model, they were very similar to what you see in the embryonic heart and liver. And this has potential opportunities for application in different areas of fetal development toxicity, which is a critical segment in development and reproductive toxicity studies. And this is important because heart defects are the most common congenital anomaly, happening in 1 out of every 1,000 babies. Of course, how much of this is due to drug toxicity, we don't know, but it really is a significant congenital anomaly. So that's the first, the second one. Now, next is neural organoid model. And this is quite interesting. It's another very recent organoid work that was published. And this neural organoid was derived from human-induced pluripotent stem cells. And the group actually validated this model for folate receptor, including its role in neural tube formation. So they use organoids with knockdown and knocking and knockout on neural organoids for the folate receptor to really show the fact that this model could be used to recapitulate what we know about neural tube defect in babies, which is a very serious early birth defect that is also very common. This has potential application for use in preclinical neural tube defect modeling, which currently is done even imperfectly in rat models. Then the next is 3D palate fusion model. In this model as well, they use 3 cell types, epithelial cells, mesenchymal cells, and endothelial cells, co-cultured to mimic palatal fusion. And this was shown to be sensitive to the disruption to the process by chemicals. And it also helped to review some of the signaling pathways that are involved in human palatal development. Cleft palate, we know, is a major structural defect that is also very common. So this has potential application for use in this important area. So the third aspect that I want to touch on is real-world data and real-world evidence. There is an ongoing study, which is a collaboration between the University of Maryland and the FDA. And what they do in this particular study is to query real-world data from electronic health records. And they use that to identify drugs that are most commonly prescribed during human pregnancy. And then they go on to use pregnancy PBPK models to characterize exposure. And they look at the data from the modeling exercise to prioritize drugs that need to be studied in human pregnancy for clinical studies. So I saw this, I thought this is a really interesting way to make better use of real-world data. And the second approach, which also involves the use of real-world evidence, is to query real-world evidence data. For instance, Sulaima, whose picture you see there, is a member of my group, who is doing a PhD querying data from the FDA adverse event reporting system. And what she's trying to do is to identify drugs with safety signals. The safety signal does not necessarily mean that the drug is responsible for the outcomes that she's looking at, but it simply shows that for this particular drug, it seems to be a lot more reported for this particular outcome than all the other drugs on the platform. And what she intends to do is to then take these drugs with positive signals and use our organoid models and our placenta-on-chip models to query them and see, does this have any impact on any of these organs that we're modeling in this way. And what we hope to achieve is to be able to inform prescription information changes, especially when this is taken to a higher scale. So this is where we currently are in the way we conduct these studies, like I showed you previously. But this is where we hope we'll move to. Take these studies away from the end of drug development and bring them right to the front. But as you can imagine, this will involve not just a placenta-on-chip model or a neural model, but a combination of different model systems. Because one of the key advantages of animal models is that you can look at the impact at a whole organism level. It's impossible from in vitro modeling perspective. But if we can bring together a battery of assays, maybe we can answer key questions. And this can be integrated with PBPK models to use the clinically relevant exposure levels in this experiment. And of course, integrated analysis of the huge, vast amount of data that will be generated from these types of studies. So this is where we need to go. And I really hope that all of us will begin to think in this way and collaborate with relevant scientists in other disciplines to develop these models and to use them for the purpose that we're interested in. And I'd just like to acknowledge my colleagues, funders, and I thank you for your attention.